So, good afternoon, everybody. I'm Angelo, and here's João. We are presenting today the the title of our project is Investigation of Extracellular Stimulation in Oscillating Networks. So, the statement of the problem is the electric stimulation used in different pathologies, such as Parkinson's disease and other motor diseases, and in different formats, so such as TDCS, TACS, or DBS. These ones are transcranial, and these ones it's implanted. So here is current, direct current is here in alternated current. Uh, it's used, but the mechanistic explanation of the effects of this type of stimulation is yet not clear. So there's there is there's a need to investigate this more. So in our project, we investigate the role of periodic stimulation in an oscillating network, evaluating frequency locking and synchronicity and variability of spike dynamics. So our methods consist of constructing a network composed by 100 Hodgkin-Huxley neurons, which 80 excitatory and 20 inhibitory. There are connections each to all of the cells and inhibitory neurons to excitatory neurons. And the network, it's working? Yeah, it's working. The network is said to oscillate by itself by a given on a single stimulus. So it have a natural frequency. The stimulation, the characteristics of stimulations is sinusoidal and constant amplitude, position, and time interval that it occurs. Here, you are only evaluating the varying the frequency of this stimulation, and this was the most challenging part for us because most of the neuron simulators, neuron-based simulator environments, does not have any specific methods, a specific tool to implement the extracellular space, and so we have to construct our code for this. For analysis, we use raster plots to evaluate spike train LFP and a spectrogram of LFP signal, and synchrony. We use Kuramoto order parameter, as we've seen in the previous presentations, and we also use the metrics of variance of ISI, so interspike interval variance. So here's our results. As you can see, when we apply 7 Hz stimulus in the network, it starts to, there are sometimes that no neurons spikes when the stimulations is applied, and after also this happens, so there is a quiet moment for all the neurons that don't spike, and in 40 Hz, 14 Hz, so we also see like that, but more with level variability and more coherent spiking between all neurons. And also when we stop the stimulation, we can also see that some quiet moments appear. And when we get to 25 Hz stimulus, the network synchronizes almost perfectly. So it gets very well, like a barcode, almost, the raster plot. So this happened. And with 50 Hz stimulus, we see as synchronization, but there are some strange things happens. Some neurons do not spike during the stimulation. So as we can see, the LFP spectrogram in 7 Hz stimulus. Oh, this we cannot see here, but in 7 Hz stimulus, we clearly see that it is presented. This is present in the LFP signal. There is a very soft line here, if you believe me, around 25 Hz. So yeah, it's it's not able to see here, but there is a very yeah not not well pronounced line here. So that's the frequency, the natural frequency of the network. And then when we get to 14 hertz stimulus, the the frequency band here increases. So there is more there's more frequencies presented when the stimulus arrives. And with 25 hertz, we can see a well-pronounced frequency of the network and increasing the band of network of frequencies that the network exhibits. And also with 50 hertz stimulus, the band of frequency amplificates. So there is these results.
Okay. So here we measure we measure synchronicity using Kuramoto order parameter. So here we have some ugly plots, but I'm trying to explain each one. Here in the left we have a plot Kuramoto order parameters in functions of time. So for each time we measure this the synchronicity, right? So as we can see, when the frequency of external stimulus reach a value of 25 hertz, we see that in the moment of when this external stimulation is applied, we can see the Kuramoto other parameters reach a value of 1. So it tells us that okay, now we are observing synchronicity, and above of this value, we can see a stranger behavior here. Uh, we can see that the synchronicity is not achieve a value of 1, but decrease as we increase the the frequency of external stimulus, right? So this plot's very ugly. So we try uh, to measure this in a other way. So here in the left we have basically the same plot, but we take the mean value and its standard deviation. So as you can see below of the natural frequency, we see that we can observe synchronicity, but this value is closer to 0.5. And as we increase the frequency of external stimuli, we achieve a value of 1. So, and the error bar decrease, right? It's a kind of small er, error bar. So here above of this value, uh, we can see a kind of strange behavior like that we observe that here we see that the synchronicity decrease but the error bar increase. So that's why this behavior, because there is no well-defined value of synchronicity. So that's why you see this large fluctuations. And in the right, we plotted the interspike interval variance. So here, uh, uh, as we achieve a value of 25 hertz, we can observe that the variance uh, reach a value of 0, that we don't observe any variability in spike dynamics. So, and above this value, we can see that this measure continues. It's it's closer to 0, right? So I think we can observe here in the in this error bar and this measure this behavior, right? So uh, and here is the conclusion that we did. So so the question is what is what is what is modulated by external stimulus in oscillating networks, and the answer is the this synchronicity, right? So we don't convince that this is a kind of resonance phenomenon, because it's very similar, but we don't convince it's is this resonance, because we're talking about synchronicity and etc. So that's it. So, and we can.
So for frequency locking, we see that the network gets frequency locked when applied stimulus or when the applied stimulus arrive in the network, mostly in frequencies higher than 7 hertz. And after the stimulus end, the networks go back into the natural frequency, most of the part. And that's that's the work that that we have done. So the future directions now is to explore the we hypothesize that the network would have a relaxation time in the Kuramoto parameter or order parameter. So, and after the stimulus is over, so as we can see here, this occurs when because of the same seeds of all simulations, but after the stimulus end, there is something weird happening here. So that may be something uh, important to explore. No, let's go back. Yeah. Um, can also we can also maybe see the altering of the dynamic state of the network, and we can also alter the model parameters such as neuron density and different morphologies and all of that. And that's it.
So I'm sorry, I think I missed the point, but when you explained that you apply the perturbation to the network, you apply for both populations, the excitatory and the inhibitory. And do you guys have any intuition what would happen if you apply only to the inhibitory population and only to the excitatory population?
It's a good question. I don't know what to expect.
I don't know how to, to answer this, but in the the motivation behind this project is to simulate a neural tissue, and what if what happens when a naturalistic like simulation, so we can replicate like TBS or TDCS, altering the network dynamics. So I don't know. It was it will be like that. Yeah, yeah. Mm, mm. This the spectrogram, I don't know how to answer exactly your question because the spectrogram from okay. this is not for the raster plot, this is for LFP. So, yeah. Yeah. I didn't see that. This is important. Mm-hmm. Uh-huh. Okay. Mm-hmm. Yeah. Okay, thank you.
Okay, I can say. I think it's kind of easy to learn NetPy, and we need the time to think how to use in a project like that. Uh, like now, I know how to uh, to do a model using a package that the goal is to understand the dynamics in a spiking neural network. So I think actually I don't I I didn't know how to use NetPy. I didn't is I I didn't build a spiking neural network. So here I learned in this type of simulation. So it was very nice for me. Yes.
And for me, it's also like that. I learned to use NetPyNE because before knowing NetPyNE, construct neural networks by constructing each cell. Now NetPyNE is very helpful, and the analysis is very new to me. All the last ones, so the Kuramoto analysis is one of the things that I learned. Okay. Okay.
So I think Thiago and Diogo, Fernando and Diogo, he's helping us to do these simulations. And I thank you, Rock, for the school.
And Valerie also.
Yeah,
He
yeah,
also
great.
Valerie.
And now it's working? Ah, okay. Okay, so And we proposed to this project that we were going to do a mean field transformation to try to explain or to try to understand better this transformation of a population code into a place code in inhibition-dominated networks of spiking neurons. And we do already a small adaptation in this title, and we are not going to talk a lot about this mean field analysis because we didn't really finish it. Okay, so basically, when we started this project, we wanted to understand how a certain phenomena in sensory systems. And Mato, for example, he talked about this process called orientation selectivity. Also, Ito, a lot of people talked about this orientation selectivity. It's a pretty famous phenomena, actually. And this emerged when they, on Ruben Wiesel, that received the Nobel, they recorded those neurons in the V1, and they found out those neurons are selective to different orientations of a luminous bar, forming those tuning curves. And basically, some researchers started to think, how could this selectivity emerge? Because in the LGN, they were not seeing that, but when they were in the visual cortex, they were seeing these tuning curves that are narrow and specific to each stimuli. And one of the proposals at this point was based on those experiments that they found out that in cats and macaques, they have neurons that answer preferentially to stimuli that have similar preferred orientations than they have. And based on that, they proposed that probably you could have this emergence of orientation selectivity if you had a Mexican hat type of model, for example, that was also told here at LASCON. And basically, those models, they consist in you are connected with things that are close to you, and you also inhibit things that are in the long range, so you also inhibit things that are far away. And based on this kind of model, they proposed that could arise this emergence of orientation selectivity. And this was kind of well accepted, not this much because you have neurons both being excitatory and inhibitory at the same time, so it doesn't respect the Dale's law. But it was well accepted because they were finding this kind of maps in those macaques and cats. But then, when they when they started recording from other animals, for example, mice and rats, they're pretty frequently studied in neuroscience, they found out those unstructured spatial patterns in which neurons, they don't have any preferred feature to be in space. And basically, to understand this transformation, and they also have this emergence of orientation selectivity, so they have this sharpening of tuning curves. And to understand that, several different researchers have been proposed in models. One of them, Mato did his invited lecture on it, so you have a feed-forward feature mixing model, and in which you can have this sharpening of the tuning curves. But also, not only that, you can also have that in balanced neural networks. And that was actually first proposed by Wrzweicki in 2012 in Hansel. Okay. Yes. Okay. Before that, one single thing that is important here, you can have those two things, but there is a specific condition for that. You have to be in those balanced neural networks. So you need balance between excitation and inhibition to have this transformation and this emergence of orientation selectivity. And basically, we wanted to apply those things that we talked about here a lot into a different problem. And basically, we were trying to understand this ITD, that is a spatial authority cue, in which you can estimate the position of a sound source in terms of the azimuthal plane, based on the time difference that it takes from the sound coming from the source to arrive to your right and left ears. And you have, basically, the thing that is interesting here is that you have different animals, and those different animals, they have different ways of representing those ITDs. So, for example, in birds, they have those type of code that is called place code, in which you have like those super specific tuning curves that are narrow and specific to each of the ITDs that you're presenting. And in mammals, by the other hand, you have those super broad tuning curves, and those tuning curves, they answer preferentially to things that are in the contralateral M field. And this basically means that if I'm recording from a neuron that is in the left hemisphere, it's really probably that this neuron is gonna answer for things that come to my nose to the left. And that's basically that. And the interesting thing here is this, that this information, this neural representation is transformed between the inferior colliculus and auditory cortex. So here we are talking about gerbils, because there's also a transformation in birds, but they are well studied. So we are trying to understand something that is not well answered. And basically here, you have in the inferior colliculus this population code that we talked before, and it seems this suggests that in the auditory cortex you have this transformation to a place code codification, because here you have at least it's clearly that we don't know really about the sharpening of the tuning curves. We're going to talk about the two characteristics of this process of transformation. But here we have clearly the distribution of peaks. So it's conversion to something that is almost uniformly spanning all the ITDs with those preferred ITDs here. So that's it. And okay, so basically thinking about that, we were trying to understand this transformation between a population code and a place code. And to do that, we implemented some simulations in NEST to investigate these random balanced networks as a mechanism for this transformation. And we used a model from ModelDB on rotation selectivity. We adapted this model to have the features that we needed to study ITD transformation. And it also was a model in NEST too, so we had to do a lot of different modifications to the model to work. And the second idea that we had was, after having this model working, to apply a mean field analysis to understand the mechanism underlying this sharpening and the distribution of the tuning curves. And actually, we have some problems with NEST when we were trying to adapt the code and do everything to make it work. So we lost a lot of time in this step, and then we could not really finish our mean field, but we've thought about it, and we're going to talk in the end. But before going to the next slide, I just wanted to say that the two important things to have this transformation from a population code to a place code is this sharpening of the tuning curve. So we have a broad tuning curve there that become a specific tuning curve. And also this distribution of peaks. So we want those peaks that are extremely biased to uniformly span all the ITDs. Not uniformly necessarily, but almost uniformly at least.
Yes. So here's our network. So we have a excitatory and an inhibitory population receiving inputs, Poissonian inputs from three sources. The IC is because we are thinking in this relation, this comparison between auditory cortex and visual. So in visual cortex, it's common to think that LGN connects to V1. So we are thinking, okay, so in auditory, we'll have IC connecting to A1. But this is just a Poissonian process. I'm trying to give some biological context because of these connections. But we could finish the mean field analysis, but we did the network thinking about that, because in mean field, you need a large number of connections, but this large number of connections must be really smaller than the number of agents in your network, the number of neurons. So, which is, it's called K for the connectivity and N for the number of neurons, where K should be way smaller. So we have a K of 10% of connection for 12,500 neurons. So we try to approach the modeling using the mean field approach, at least for this. And our inputs are a bimodal distribution. So we have minus 160 and plus 160. So it's a bimodal distribution, two types of orientation, we can say, in this manner. Okay? And the relation between inhibition synapse, the strength of the inhibitory synapses, which are stronger, also thinking about the mean field.
And so just to be a little bit more clear, what we do is like this inferior colliculus, how we model these inferior colliculus is that those neurons in the auditory cortex, they are just sampling from one of the populations that we have, so or the one that is the left hemisphere or the one in the right hemisphere, and then we just have like the preferred orientations being minus 60 or 160 or minus 160, and then this Poisson process have this the height defined here. based on the neuron that it was taken from this bimodal.
So here we have in the raster plot, we have a population receiving with a strength coupling of 0.01 millivolts and receiving just one type of stimulus. So it's minus 160, and we can see half distribution of half of the neurons has increase in the firing rating, and another half it's slow, so they are responding to our process. And in the left image, we took one neuron from this raster plot, neuron 8771, and we plotted his firing rates according to the input. So from the stimuli, right. So in the lower part of the plot, we see there's a higher concentration of spikings, so which is the preferential for this neuron. And as we go to the positive side of the stimulus, it spikes less. So this is how our network is responding to our stimulus, so we can plot the tuning curves.
Yeah, so based on the graph that we showed before, we calculated tuning curves of the neurons. We did actually 10 trials in which we have all the stimuli from 10 to 10, from minus 106 to 106. So to run a full experiment of those, we took around 5 hours and a half to each of the EPSPs that we are using. And basically, after we extract those tuning curves, what we can see, the tuning curves up here, is that there are some important things to say. The first one is that if you take a look in the epsilon axis, you can see that we are decreasing the firing rate as we are increasing the PSP, because we are in this inhibition-dominated regime in which we have really strong inhibition synapses. And besides that, we can also see that as we increase the PSP, things become messy here. They do not see like the input tuning curve that we input in our network. And also that we have this distribution of the peaks. Yes, we have this distribution of the peaks according to the strength of the coupling that we are inputting in our network. And in this case specifically here, it's important to mention that it seems, we didn't do like the analysis, but it seems that we are losing the stability of our network. It's becoming something really messed up.
Yeah, it could suggest, be careful, I'm not affirming, but maybe a loss of stability of our network as we increase the coupling strength. Yes, and also the scale on the y-axis of the peaks is also important. So we kind of see the distribution becoming uniform as we increase the EPXP. So this is also a thing to investigate doing mean field. We can find a solution, look for the fixed point, and see what's happening in the relation of the coupling in this loss of stability.
And yes, so basically, I said before in the objectives that we need two things to be able to do this transformation from a population code to a place code. Of course, we're not going to do a full transformation, a perfect transformation, but these two elements are important: the sharpening of the tuning curves and the distribution of the peaks. And to be sure that we are having that in our network, what we did is that we calculated selectivity index, or the sharpening index, to know how more sharpened it was, the output than the input. And basically in our input we have, so this was our input, right? And then we calculated this by applying the 1 minus the circular variance. And based on that, we obtained this value here for the input tuning curves. And we calculated that on the, on all the neurons of the network. And based on that, we had this distribution in which we can see that for EPSPs, for example, of 0, 0, 1, and we have already a selective index that is, we have a sharpening of the network, so the mean is already more to the right. And as we increase the PSP, it seems that we can shift this mean a little bit more, but at the same time, by doing that, we broaden our tuning curves, not our tuning curves, but we broaden our sharpening indexes, so they become more distributed. Yes, so that's it.
And to extract some information between those distributions, we calculated the Kullback-Leibler divergence between the distribution of the peaks and a uniform distribution. Because we saw that as we increase the coupling, the distribution are kind of become uniform, so we are trying to see what happens. So we calculated the divergence, and we saw again that as we increase the coupling, this divergence tends to 0. We didn't have time to evaluate the interval between 0.01 to 0.2 to see how this decreasing occurs. So we couldn't do these calculations, but looks like the increasing, it is becoming uniform. So we kind of guess this is what happens. So a brief summary. So we saw the transformation between populational code to place code emerging from this type of network, and some sharpening also occurring according to the relation with the strengths between the synaptic strength. Okay.
Okay. Also, something that we saw those things depend on the coupling strength and both the sharpening and the distribution of the peaks, but also we kind of seeing those results that we are more sensitive to variations in smaller PSPs than in large ones. And yes, so those results, and also we saw that this decreasing in the Fahrenheit as we increase the strength. And those results, they suggest that actually this can be a helpful mechanism to this transformation, but probably not the only one, because otherwise we're gonna kill our network with the Fahrenheit. They're gonna go to 0. So probably we have something else happening here to this transformation, to convert to this uniform almost distribution. And that's it.
Ask your one, then see if there is time.
Yeah, and I'm used to work directly in data, so learning nets, modeling networks, it was all new for me. So it's the main part, and also work with selectivity orientation that day.
Yes, to me was actually some things were nice. The first ones that are no, not this one, not yet. No, okay. Yeah. First, yes. So Vinicius is really, he knows how to calculate the Kullback divergence, for example. I do not. So he let me try to implement it before he did, so I could understand what's happening. And we found out that in SciPy, for example, they do this in a weird way. So they they do not have negative values in the Kullback divergence. And then the value that you obtain when you use SciPy is different from the one that you would obtain for the definition of the divergence. So we found this out because of that, and it was nice. And also I found out that there was a bug in Nest, and I found out when Hans was here. And because of that, I had already tried DAPSP 0.2 in my master's project, so I kind of knew that this should happen, but we got completely different distributions because of this bug in Nest. And after coming here, it was super important for us to discover this bug and be able to fix it as well.
Your idea is that the switch from the place to the population would be the change in conductance?
Changing what?
Would be the change in conductance. So how would you explain the transformation? Okay, you can get one regime or the other, but then if there has to be some kind of transform,
Not necessarily. Like they are. Pardon. They are projecting to the this upstream area, and what we get is because you have a random connectivity, for example, so you're always shifting a bit your tuning curve. And I think that in this vector, for example, that's what we're trying to do in the mean field. So you're always shifting a bit the preferred simulation that you have in the input, and because of that, yeah. Okay, perfect. Sorry.
Okay, we are Tomás and Gustavo, and today we are talking about the paper from Spracer and Kumar that talks about studying the spiking activity that produces emergent spatiotemporal patterns in like inhomogeneous distribution of the connectivity in a grid of neurons. So, like we took the base model that it's from Model DB, where they basically what they talk about is like how in the literature from this paper to and going like before, the most of the spatiotemporal patterns that they were generated, they were by tuning the model by hand or by like training the model by supervised learning. So what these authors propose is create a generative rule that resembles more some more biologically plausible way of generating these patterns. And why are these patterns important? Is because they are, according to the literature, they are being displayed on the cortex, the thalamus, and the amygdala, and they are relevant to the like function of specific areas. So we are going to explain how do they implemented the model. So the original model taken from ModelDB, and it's on GitHub from the authors. It's made on Nest 2.0, and one of the like intentions that we had to work with it was to move all the code to Nest 3.0 and take this random rule that makes this generative rule and modify it to add adaptive synapses, because they in the original paper they use like constant wakes, test the resilience of the network against the loss of some nodes, and at the end from the idea from Professor Battaglia to try the stimulus input on the network. So to understand how do they make each of the like, how do you say, like conditions of where they test the connectivity, basically they use as control, they make a grid of 100 by 100 neurons. Oh, sorry. 100 by 100 neurons, and they from a like from a they pick random directions in all the degrees and randomly assign a direction to each neuron, and in all cases each neuron will project 1,000 axons to the nearest neighbor. So we have this random case, we have the homogeneous case that is the most simple to understand, that where all the axons from the neurons point in this direction, and the pairing one, that is the most interesting one, that is derived from a distribution that was like made in the 80s to generate patterns that look biologically plausible. And what is interesting from this distribution is that it makes clusters where the clusterized directions of the neurons are like more pointing in the same direction into the inside of the cluster. So that's what it looks like, the Perlin distribution, and you can make it coarser or finer depending on the index that is assigned. So in this case, all the neurons inside this blob will point to each, connect more to each other than to the others, and that's what the colors mean. So we here have an example of how the will be the spatiotemporal patterns that emerge from the Perlin pattern, because it's the only that generates these patterns. I mean, when you have correlated activity in some areas, you generate these movements here in the Perlin pattern that go like this. In the symmetric and the random, the symmetric case, I didn't mention it, but it is where the 1,000 axons of each neuron points in all directions. So one here and there and there and there. The random is the 1,000 in a random direction, and the homogeneous are the ones that are going all the same in that direction. So the authors finally conclude that the Perlin distribution resembles more the activity of the spatiotemporal patterns that are exhibited mostly like compared to the amygdala. And here we have the same patterns that were shown before, different coarseness of the, different coarseness of the Perlin distribution. So that's coarser and that's finer. And that's the quantification of the weights on these static synapses. And we're not going to show how in the adaptive case it works.
So we chose to look at the effective weight of the synapses when we add the synapse dynamic to see how the structure of the network was changing. And we also look at the PSTH because it was a quantity that we found that was quite similar for different partners with the same type of adaptability. So we implemented the adaptive synapses. And what we see is that if we implement through all the neurons in the network, the system gets to a state that is hyperactive. It's has a lot of waves traveling all around. If we do for all the synapses, and the weight distribution goes really further into the excitatory range, because in S, the difference between synaptic excitatory synapse and inhibitory synapse is the sign of the synapse. So the weights go further into the acid, and the firing rate gets this shape with a lot of waves independent of partner again. If we choose to do it only on the excitatory synapses, the adaptation, we see that the clusters, they travel for a bit and freeze because of the interaction between the synapses and the inhibitory that is still there and much stronger than the excitatory. Because when we look at here the last one, we look at the firing rate, we also see that the system stabilizes in a constant firing rate after some time, and the effective weights are much lower than they were in because this here is the inhibitor that are static, and the one that was adapted is only this. So we follow Professor Battaglia's suggestion and try to put a stereo input, And in the static case, we see that the response to the input is really high. The clusters get faster and starts to appear more spiking. And we can see here that here in 1,000 milliseconds, there is a increase in 1 picoampere of current. Here we have a 10 picoampere, and here we have 100 picoampere. So every time we increase the input, the system reacts. While in the all adaptive case, we don't see that. Here we have a change from 0 input to 100 picoampere input, and you can see change anywhere. In the other case, we see a intermediate situation that the system doesn't adapt through small inputs, but if you put large enough, the clusters grow. Now, no, yeah, there. But this is we are applying an input to all the network. So after that, we try to do it in a localized region. So for the case, the static case, it doesn't really talk much if you put on a region in between different directions. In the case for the excitatory one, you don't see a response. You can see the difference no matter what you do. But when you put on the static case close to a region where the clusters flow, it inhibits the movement of clusters in that direction of them. Here, you don't see any clusters going downward. And it can even create clusters going in the other direction, but they have a different behavior than the ones that naturally arise from noise. Here, the same thing for the other configuration, but now if you put the input into a region that already has a cluster, he is the only one to get bigger, which is expected. And if you add the input before you put the noise in the system actually reacts to it and creates a wave partner wherever you choose to put. And then if you turn on the noise again, you don't see any change. And if you have the noise before the input, you don't see the input. So yeah, after that we try to remove nodes to see what happened to those partners. And in the case for the static one, you can still see for a good range of, here we took 10% of the network, here we took 25%. But for 50%, you can't see anymore the traveling clusters. And in the all adaptive synapses, you can see those partners even with half the network gone, half the excitatory neurons gone, and the behavior is still the same, the fire rate. But and to finish when we do the same for the only excitatory synapse case, you can still get clusters, but they are way more improbable. They take a lot more time and activity to be generated. And, well, as conclusions, we have the addition of plasticity can freeze the clusters in regions of space. If you put only on the excitatory networks, then can generate really resistant kind of partners if you put in all the synapses. And all the three of them could resist even if they lose a great amount of neurons. And as perspectives, we could try to take a look at the function network generated by those partners and measure entropy of the partner activity. And if we would try, for example, to make 2 layers of these networks with different partners, how would they interact? Are questions we could try to answer. That's it. Probably the first one, the waves are traveling, so you can send information from one place to another, like the static sense. Yeah.
Which is the most controllable
Oh.
regimes?
So the one with excitatory synapses adaptive, because you can. Would be the case with excitatory synapses because you can create clusters with. Yeah, I mean, the when I saw the previous group presentation, I thought maybe if their system was spatially organized, could something be seen in the direction preference of the neurons. I don't know, maybe.
So when you showed that you apply the input to the network, and then you
Which one?
this one, I think it was the first example for you disconnect nodes. Yeah, so that one that was before. Yeah, so here, for instance.
Okay.
So you say you're plotting the firing rate as a function of time, but you are applying different inputs for each time. Is that correct?
Yeah, yeah. And the scale here is mass, but the where we were looking at is the shape of the curve to see the.
You have 3 inputs there.
Here we have 4. When we put 1 picoampere, you don't see any change in the behavior. But when you put 10, you can see a bit, and then other.
And the input is there for the whole time. You're just changing the
Yeah. In this case, yes.
off.
Goes back down for this case, but not the. No. Yeah. But does in the with the one with adaptation. Any more? Well, I learned to save to save videos on Python, but I also learned S because I didn't know about all those types of programs that can help you create a lot of different stuff. I used to just code by hand, I think.
Yeah, me also got like low experience programming and I'm really new to computational neuroscience, but I really like the idea of like being able to tune the model and have a little bit more time and see like which patterns are we trying to mimic in at the tuning of our parameters will be nice. I mean, each simulation took like kind of around 2 or 3 hours, so it was kind of like hard to pick and decide on which like direct direction move the tune the parameters. But it's a good start, or at least it was interesting. Yeah.
Just was like to thank Professor Hans that helped us a lot with making the problem. Yeah, we had the NES2 code, but we tried to to replicate with the some new functions that are available in S3. We are unable, and hence say that it, to be done will need a lot more work. So we translated from 2 to 3 the code instead of making a new one that does the same.
Specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
It's okay now? All right. So good afternoon. My name is Graça. My partner is Maria. And now we are going to present our project, which the title is How Connectivity Between Neuronal Populations Impact on Synchrony. And this is overview of our
project. So what motivated our project is actually a little bit of my own background, and also your lectures, Professor Battaglia, because we showed us how functional connectivity can be indirectly measured via synchrony between populations. So I feel like the most common approach in scientific literature is to use is to measure functional connectivity in a non-directed way, so you can know who is sending information to who, and usually model-based. But you can also find something that would be, for example, Granger causality, that you can infer the direction of the information. And Dr. Battaglia also showed us that you can do something that is model-free, and perhaps that also can increase our knowledge. But I'm focusing here, which is the more, I feel like, established type of analysis, and you can easily find it in packages such as Elephant, which is what we used. And usually for these you use analysis such as coherence or phase logging value or pairwise phase consistency, which measure how similar are rhythms across different populations or across different areas. So we know that functional connectivity can be measured this way, but sorry. Apparently, we wondered perhaps we can show something about structural connectivity as well. We wondered, I got this sort of idea from a lecture from Arndt, and he very briefly told us about this paper. And this is a very highly cited paper, actually. They used neurons and in V1 in mice, and they did pairwise correlation in this in some of the cells. And basically what they found is that the most of the cells are weakly correlated. So you can see like this is skewed, like pushed to like the axis. So most pairs are weakly correlated, and actually most of the connections have a very weak amplitude of the EPSP. So mainly there are very little strong correlation, strong response correlations and big amplitudes, but they play a significant role in specifically in V1 in apparently neuron selectivity. So you can see here, for example, in H, that the response correlation is it's like correlated. No, it's yeah, it's positive. Thank you. It's positively related to response correlation. So pairs that show a strong response correlation tend to have a higher connectivity. They are more likely to be connected. So as as both me and Graça, we never modeled anything, entire lives, we felt like this could be a broad enough and a simple enough project that we could do in this space that we had. It turned out that it did not, but we tried. And basically our goal was to have an intuition, create a model that has an intuition about how structural connectivity can influence the coherent activity between neuron populations. We thought about it and we created our networks thinking that they would display oscillating behaviors. They did not. And after that, we wanted to implement some of the synchrony measures that we learned about, especially during E2 lectures. When we thought about this, we implemented this in Nest. We thought that we could perhaps use synchrony measurements in LFP as well, but turned out that Nest does not allow it very easily, so we only worked with synchrony measures in spikes.
So as Maria has said, we wanted to implement this population in Nest because the two of us want to learn how to simulate with Nest. And the population that we have simulated is shown here, where we have 3 populations of neuron, 3 big populations, the population A, B, and C. In each population, we have 20% of the population being of inhibitory cells and 80% of the populations being of excitatory neurons. And we have connected these populations unidirectionally with the probability of connections between population A and B being higher than the probability of connections between population A and C, and also the connections inside each population are higher, have a higher weight than the populations between the then the connections between the populations. So the model that we have implemented in NEST was the current-based leak integrated firing neural model with delta-shaped postsynaptic currents that is given by the equation, where the parameters that we have used was these ones that are the default of NEST to this model. See, and here are some of the model parameters, another parameters that we have used, where we have tried to give a higher weights connections to the inhibitory connections to try to do our network have oscillations. And as I have said, the probability of connection inside a population was higher than the probability of connection between the populations, with the probability of connection between population A and B being also higher than the probability of connection between populations A and C. And these three populations were connected only using the excitatory neurons. Only the excitatory neurons were connected. You pass the simulations, these are another parameters of the simulations. So here is implement the neural model with the default parameters, only consider the different probabilities between the connections of the populations. Here we have the raster plot of each population. Here is A, B, and C. And here is the potential of the membrane of each population, only showing the case of the excitatory neurons. Here, the next, yes, sorry. And here is when we have implemented noise using a Poisson generator with frequency of 50 Hertz. And we could see that we have a little difference to the previous one and between each populations. And also the next one is when we inject a DC current of 10 picoamperes from the time 0.1 millisecond to this time in our model. We have this. Yes.
So those were the starting parameters. I tweaked them a little bit to make their hazard plot seem a little bit more reasonable than they were. And so I tweaked. And in the end, for example, I wanted to use some of the notebooks that the tutors left us. So this is a notebook that Ito provided. So I used the elephant library, the statistics about spike trains. I actually forgot to change that. This is not trio ID, this is neuron ID, I'm sorry. And this is the pyramidal neurons of population A. So even though we gave a higher weight to connections, to inhibitory connections, I actually cannot see any sort of oscillating behavior here. And yes, so, but thank you. And also I tried to implement a peristimulus histogram. So here in population A as well, which is the population that received the DC current. So here you can see, probably due to the input, the big firing rates. And then after the input ceases, the population sort of restores. In the PSTH, I can sort of see an oscillating behavior very lightly. Not anything like some of what you brought today. And actually, I don't understand what went wrong, because, yes, such is life. So population B, which is the most strongly connected to population A. This is the raster plot. And one of the things that surprised me when I saw this raster plot is that even though I did not put an input in this population, it seems that like the firing rates of population B and C are actually higher. And so this is also the PSTH of this pyramidal neurons. And the same for population C, which is weakly coupled to population A, with also PSTH. What when I was computing this, what I found unnerving perhaps is that the PSTHs of population A, B, and C, they looked exactly the same to me. So I imagine that, and perhaps I still feel like I must have made some mistakes. But overlaying them, you can see more or less what I mentioned about population A, even though is the one where we injected the current, having the lower firing rate in comparison to population B and C. And yes. Also, using Ito's lectures and notebooks that he left, we computed the probability densities of the different interspike interval distributions for the different populations. So we can actually see that populations B and C are not too different, and they are shifted to the left, and population A is actually shifted a little bit to the right. So I feel like populations B and C display a behavior that is more similar to, it's not Poisson, I think it's gamma distribution that it's like this. I don't remember. I think Poisson told us about this, but I not grasp it. So population A is a little bit different. And finally, we tried to implement some of the synchrony measures using elephant. And well, it did not happen. Basically, so this is the cross-correlation analysis. And what we do is you plot like population A versus population B or population A versus population C. And I would expect that, for example, the synchrony measure of population A and B to be higher than A and C. And they actually look pretty much the same to me. And this actually does not look like anything I would, it does not look like anything real. Especially there's a very narrow peak where there's like little phantom peaks. It looks like perhaps some kind of mistake when I implemented this. I really had a hard time with elephant, especially importing, like making my spike times data, which I got from Nest, importing them into elephant objects. I feel like I messed up that. I cannot have that messed up that. But this was the first approach that we took, and it was a failed one. And the second one was also a fail, because we tried to use complexity differences between the populations. So basically, can I just go back one? I feel like one thing that also that was difficult is that to do a proper correlation analysis, you need many trials, and we could not do that with Nest. We simply could not compute. Sorry. Okay. We simply could not compute that. So I feel like the fact that we only had one trial made this measure pretty much like trivial. And also the difference complexities. So basically we plot the probability of the CPP. In this case I use population A and B and the surrogate, and we plot the difference between them in various using various bin sizes. So this is different bin sizes, and this picture is completely wonky. It's not at all what I would expect, and I don't know if I feel like it's both. I failed to implement oscillations. I failed to make them synchronized, but when I forced them to synchronize by applying an AC current in a continuous form, even with that, my two approaches, they also did not come like they were supposed to be. So I feel like I also failed implementing them. And yes, so sorry, not a very interesting, everything did not work out. But nevertheless, we are thankful for the tutors that helped us. It was very, our first experience creating a network. So Dr. Junjito, Hans, who also helped us creating our network, and his student, José Villamar, that answered those questions. And also, thank you, Dr. Bazinski, because you and tried to help us yesterday. Yeah, thank you.
Okay, thank you everyone. So in our presentation, we would like to show you our strategy that we use to model and simulate the effects of benzodiazepine on inhibiting synapses of CA1 pyramidal cells. So just to give a bit of background, what are benzodiazepines? Well, they are a very large family of drugs which are used to treat different conditions from anxiety to insomnia to acute seizures. And what they do is basically they enhance GABA A activity by increasing the inhibiting postsynaptic current. And this way they hyperpolarize the neurons, so they are less likely to fire. And of course, this also have an effect in changes in brainwave patterns. Why did we choose benzodiazepine? Because they have a very simple mechanism of action. They bind to the benzodiazepinic site of GABA A receptors and only GABA A, so no GABA B. And when they do bind the receptor, basically what happens is nothing. They don't open the receptor, nothing happens to the neuron, unless also GABA is present. So they have a GABA-dependent mechanism of action. And this simplified a lot our work because we could only take into account the effect on the iPSC when the synapse was active. And we did not need to add any extra current to the neural model. So what do we mean when we say that we want to model the pharmacokinetic, pharmacodynamic effects of benzodiazepine? Well, basically, we want to show the effects on a system, on our system, based on pharmacokinetic parameters that, in our case, were different drug concentrations. So in order to do so, we first wanted to model, customize GABAergic synapses in order to see the effect of different concentration of a particular benzodiazepine, midazolam specifically, on the iPSC. And then see how this customized GABAergic synapses can affect network activity, mainly focusing on the beta band frequency because it's well known that benzodiazepine mainly affect this waveband. Which is indeed used in experimental, in animal experiments as a measure of the efficacy of the benzodiazepine. So what we did was we took a CA1 microcircuit model created by Ponzi and colleagues with NetPyNE. We reduced it. This model comprises 3 neuron populations, 2 inhibiting interneurons, basket cells and all interneurons, and 1 excitatory population, pyramidal cells, or connected between them, and also with recurrent connections. So the C1 microcircuit is built using multi-compartment morphological detailed neurons. So the neurons are displaced in the simulation environment, in the simulation space, according to their real dimensions and their real morphologies as taken from neuromorpho.org database. They are Hodgkin-Huxley models which embed different type of currents. They have sodium currents, 4 types of potassium currents, 2 types of calcium-dependent potassium currents, and 3 types of calcium currents. And the channels are distributed along the dendritic branches and the axonal tree according to the literature, so very detailed in biophysical meanings. And the neurons are then connected between them with Sodix Markram synapses, so including GABA A, GABA B synapses, AMPA, and NMDA. So first, what we did was to customize our GABA synapse. So in order to do so, we fitted some experimental data that we found in a paper by Baez and colleagues. So in this paper, Baez was basically recording the inhibiting postsynaptic cells via voltage camp on pyramidal cells when they were stimulating a norm interneuron to elicit a single synapse in the pyramidal cells. And they did this applying to the slices of CA1 different concentration of midazolam. And what they obtained was the changes, the increase in the inhibiting postsynaptic currents at different given concentration of the drug. So once we customize our synapse, we embedded in 2 small networks. We first built a toy model just for a proof of concept to see that everything was working correctly. And then we embedded in a reduced version of the Ponzi and colleagues CA1 microcircuit. In our case, we reduced the network to only 2 populations, so pyramidal cells with recurring connection and all interneurons. And of course, then we went to see like how our new synapse affected the beta band activity.
Okay, so since the main idea here was to model a behavior in a single cell level and see how it affects a larger scale, so a network, the first step was to model the conductance as a function of the drug concentration. And we model it as an amplifying factor of the conductance based on what we saw in the paper here. So we fitted this curve for different drug concentrations so that the conductance is now depending on the log of the concentration. We model it as a simple second degree polynomial, and we get so the different IPSCs depending on drug concentration. It changes the amplitude of the IPSC. So then with the cell modeled, with the customized cell that depends on drug concentrations, we embed it in the network. So first we have the toy network we called. This network it contains 20 pyramidal cells, 20 Olm cells. They are much simpler than the ones from the other model, but they generate this oscillatory pattern. And what we want to see here, as we explained, is a difference in the beta band. So we look at the frequencies between 15 and 25 hertz. We look at the power spectrum on that band, and what we expect to see is an increase on the beta band once we apply the drug. When we look at the results, we actually see the difference between control scenario and the different concentrations of midazolam. And when we look individually at each drug concentration, we also see the curve that resembles what we are expecting based on the way it was modeled and on the results from the original paper that describes how the conductance change based on the drug concentration. Then we moved to the CA1 detailed microcircuit. We rescaled it, but we never changed the biophysical parameters. We only changed amount of cells. So we ended up with 25 pyramidal cells, 2 Olm cells, and an external input that simulates the effect of the Schaeffer collaterals. In this case, we did, again, the same analysis. We look at the beta band. We expect to see an increase in amplitude. When we look at control versus midazolam scenario, we also see the difference in the beta band. But when we look at the different power spectrums for each concentration, we don't see the expected curve. We couldn't manage to get a good result in this. It's probably something related to, we rescaled the original model, and rescaling is never an easy task. We learned this. That's definitely something we learned. So as discussions and perspectives, the modeling of pharmacokinetics and pharmacodynamics of drugs requires like a multi-scale approach, and that's never really easy to do. Taking a model and rescaling it and using for a different purpose, it's also not a pretty easy task. So some different results that we could study are the effects of the drug in a different dimension, changing the dimension of the network and see how it goes. Looking at different scales, not just the CA1 microcircuit, but an even larger network. Looking at effects on other bands, there are some reports that show that midazolam also affects the gamma band, but we just looked at the beta band here. And the effects on physiopathological conditions as well, because one of the main uses of benzodiazepines are for stopping seizures, for instance. And so if we could take a look at a seizure network and see if it can stop the seizure. And that's it. We don't have an acknowledgement slide, but we would like to thank Valerie a lot because he helped us really, really much. Thank you. This is from the original network. It shows beta oscillations based on how it was constructed, the original network.
So then would you expect increases with the fly spin, or it was, so was this also in the original model, or was something that you added and you expected?
We wanted to see if changing just the cellular level, just a single cell, the single cell model that is embedded in the network, we can see. No. Yeah. We want to see if this alteration could show some effect on a network level, on a network scale. I have never worked with any of those modeling tools, so I think most of it was really new for me, and it was a fun experience building a model or adapting a model.
Like for me, it was very nice to finally see like something that I know, like drugs, for example, and like use pharmacological mechanism inside a computational neuroscience approach, because it's something that was very, very new to me. And so I'm very happy that at least like I could we could do something that is more related to my background inside the computational neuroscience field. And of course, the use of NetPyNE and Neuron that I never used before, and especially NetPyNE was very cool to use because it's like very smooth, very nice. So yes.
So, good afternoon. I am Joanna. I am from the physics department of UFAL, and my partner is João, and he is from NUMEC. And so today we are talking about the reproduction and adaptation of Wakon's neuronal model of mismatch negativity. So we aim to replicate the main results of the paper of the Wakong. So we are we have focus in the implementation of the model. So that's it.
So, good afternoon. First of all, I will try to explain a little bit about what is mismatch negativity. The mismatch negativity is a component registered at the EEG signals that appears when the participant is exposed to a odd or different stimulus in a sequence of stimuli. So, for example, it was first recorded at the oddball paradigm, which consists of the participant is hearing a sequence of equals of a sound with the same frequency, with equal time spacing between them, and suddenly it comes a different sound. So it is always listening a sound with a specified frequency, for example, 1,000 hertz, and suddenly a sound B called deviant comes, and we see at the EEG that it responds differently to this sound. So, for example, if the standard sound is 1,000 hertz and the deviant becomes or different, so we are increasing the difference between the frequencies, we see that the EEG response presents this bump here that is appearing when we increase the difference between the frequencies. And since this axis is inverted, the minus is up, we see here that this is a negativity in the response. So that's why we call a negativity, and it is a mismatch negativity because it appears when the person hears a different sound. So it occurs also with different kind of stimuli. It can occur, for example, when seeing something different. Okay, as I said, it can be explained in terms of the predictive coding theory that says the brain is actively trying to predict the incoming stimuli based on the previous stimulus. So we create an internal model of the sequence of stimuli, capturing the regularities at the sequence of stimuli. And from the biological point of view, different layers of they propose to the ones that advocate the theory that the different layers of the cortical for cortex are responsible for different parts of the computation. So one computes the prediction and the other computes the prediction error. So when it is responsible for comparing the incoming input from outside, from the thalamic input, and the incoming stimulus from the prediction layer. And if the prediction is wrong, it will give rise to a mismatch signal.
So about the model, we are trying to reproduce auditory cortex, and we have two columns. In each column we have the layers that here we have a thalamic input, so the prediction error layer and predictive layer connected to the memory. So the prediction error layer will receive the thalamic input. So connect to the predictive layer. If the predictive layer is correct, the predictive error layer will not show okay, and because the inhibitory population here. So the prediction of Souza and R A will consist in increase in the population frame rate. So the memory I modeled like a delay line is studied in the paper. So it's like a line that's in the time t, the first neuron spikes, and time t plus a delay, the second neuron spikes. The memory are connected with predictive layer, both A and B. So with the synaptic plasticity. Each neuron in the model, we have using the easy-cavity model, like the paper. With these parameters, we have a uniform window variable between 0 and 1. In the synaptic current are the sum of these currents.
So about the synaptic currents, we have 3 type of mechanisms. The AMPA current and the GABA current are described by similar equations, where we have this term here that is the gating variable associated with the quantifying the quantity of open channels. And this current is the total current that is passed to the postsynaptic neuron by the total set of presynaptic, in this case inhibitory neurons, and presynaptic set of presynaptic excitatory neurons here. And we have this weighted sum that gives the total GABA current and AMPA current with these parameters. And well, at the paper, they use these equations to describe the evolution of this S variable, the gating variable, but we checked at the paper from which they took it, that is this one from Brunel and Wang, and actually these equations should have a minus sign because, well, this is a type of exponential synapse, so the gating variable should decay exponentially. So this is a minor correction that we had to make. And here at the NMDA total current, it is like the other, but here we have this term that depends on the concentration of magnesium. And this term was also wrong, unfortunately. We looked at the paper, and this parenthesis here should be here, so this is not inside of the exponent. So I think this is the kind of typo we find in this when we are trying to replicate a model. And those are the 2 equations that describes the evolution of the S, the gating variable, and the X variable that is here. So here we have a saturating, if I'm not mistaken, saturating kind of synaptic mechanism. And they also, the plasticity between the memory and the layer 2 are of a kind of the spike time independent plasticity, they describe with this equation, but it is simply the normal equation for the STDP plasticity, but they have a dependence on this calcium current that is not present in the model, but they approximate it by the NMDA current. And they also have this weight, this variation in the weight when every time we have a presynaptic spike. But this part we did not put at our model because they did not specify the units of this current, for example. So multiplying this CP, I think it was 60 times this current could, depending on the current, could give a huge value. So we did not think they were clear enough for us to implement this part. So we made some changes.
So we use the NetPyNE because we think it's more easy to make modifiers in the program. So here we have the cell parameters. We use the easy cavity model modifier 2003 because it's easy to modify the synaptic mechanisms necessary to reproduce the paper. Here we have the populations. We have the thalamic A column, layer, statutory layer 4, the column, and for the others. Here we have the statutory memory of the A input. We have the synaptic weights, synaptic weights, and here we have a special weight to in semi connections. We have the normal between these parameters. Thank you.
So for the memory, as we said, we have a delay line. So there is a connection, linear connection between neurons. So we constructed this list in which the I neuron is only connected to the I+1 neuron. And here, the connection between, they did not specify at the paper how this connection is made, so we may have to make it on our own. The connection between the prediction layer and the memory layer. So since we have only we have to simulate only the first neuron at the memory layer. That's what we did here. And those are the mechanisms. Here we created two mod files to implement the equations we said before. And to the STDP plasticity, we used a file available. I think it was reinforcement learning arm that used the STDP, but we removed all the non-necessary parameters related to reinforcement learning. We only needed the STDP parameters. And the simulation we gave, this is a little bit too much to read, but the idea is that we are trying to implement the oddball paradigm, as I said before, in which the stimulus are of the form A or B, and they are independent. The A comes with a certain probability, in this case 90%, and B comes with 10% of probability. So the stimulus will almost always be A, and sometimes BB. So we expect to have this kind of response to the difference of the stimulus. We had 150 stimuli during 30 seconds of simulation, and with a interval between the n of 150 also. So about the results, this was the expected results. It is really pretty, but we did not obtain this. Just okay. Can I continue? Okay, okay. Here we have the memory spiking. First, in this case, we have the A stimulus coming and the thalamic input, the thalamic cells spike. But since the prediction was that it was a sound coming, this prediction error layer will not spike because it will sum the signal coming from the predictive layer with the thalamic input and will not spike. But when we have, just remember, this was what they obtained at the paper. We will show our next. When the B sounds come, this was not expected, so the predictive layer spikes because at the column A because they were expecting the sound A. But here we have the negativity because of this input, negative input, summing with this one. Thank you. When we have the B stimulus coming, it was not expected, so this column, the prediction error will be huge because it was not expecting this sound coming. This expected sound was A. So the sum coming from here and here will be a big spike. This is the mean activity of the population, and it will pass this information from here into the memory. So yeah, and this is what we obtained. That was not pretty at all, but here we have the A stimulus coming, and as expected, this population does not spike. And one spikes because it is related to the prediction of the incoming sound. But when sound B comes, here we have the prediction error population spiking because it was not the expected sound, but this information should pass to this other layer because it was not expected, and this should not spike. So, I mean, these two layers are behaving as it was expected, but this one is not behaving as expected. So we think there could be some reasons for that. Firstly, we had to implement the properties of connection between layer of prediction and memory by our own, so maybe the way they implemented makes a difference. And as the other group said, also we had to shrink the population. They used, I think, 400 memory neurons, for example. We used only 120. And maybe we had to adjust the weights mainly because of the connection of the layer 2 and the memory so that it could work. And this, I think, is less probable to be a possible reason. But the plasticity mechanisms, we made some changes that could influence the result. So that's it. And we thank especially Valerie because he helped us to have some ideas of the implementation. Thank you. Okay. I think it's this one, because we really had to make by our own. Mm-hmm. Yeah. Yeah. Maybe, I mean, at the paper they say they have inhibitory neurons at the memory, and we did not put it because we did not get how they would be added. So maybe try to understand how they use this inhibitory neurons could help.
Including one which keep code that does reproduce the figures of getting cells.
Mm-hmm.
Actually, it's nice that you thought about the reason for the thing. It's a nice replication.
Thank you, thank you.
Did you find at least 2 errors in the original formula?
Yeah, yeah. We were looking a little deep to investigate.
So, what have you two learned from your work?
You want to start? Yeah, so I think we have learned that, well, the papers can be wrong, right? I think that is really important because the papers go through review, and even with this, they can be wrong. That is a point, an important point. And actually, to learn how to use the NetPyNE and Neuron, I mean, we had to implement some mod files, so we had to learn a little bit about the code behind the NetPyNE. So I think that is, well, to work with a tool that you do not understand, I think maybe not a good idea. So if we proposed ourselves to look a little bit behind of the NetPyNE, and that may give us insight of how you to use better. I don't know, that's...
I think the same of the João. Thank you.
That's another implementation. It's kind of weird because, like, so I did the calcium. Like, obviously, calcium decays way slower than like sodium actually, but that's just digital time. But like comparing our graphs to Denver graphs, like our calcium decays way slower. And we're picking, you asked my opinion. I think our calcium decay is way slower. Because calcium decay is super slow, and so I think it's hard to recommend personally. Maybe I'm biased. I think the paper is wrong, and if we're making our project together, that's why I'm right, they're wrong. That's the best way to look at it. So when I was making waterfalls, I would call them impaired excitability. I compare them to I think possibly the presence of Denver also influences calcium decay. Maybe the zone calcium.
I have both. I have to check.
Black and white.
No, I got one. I got two. I got the white and the black. So I have both of them.
doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I
had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
Well, hello. As you can see, I'm alone because Dante, my partner, had to go to the USA to have an interview for his PhD he's applying for, so we wish him luck. As you can see, the title of our work is towards biological implementation of Bayesian inference in spiking neural networks. And the main goal of our work was first to reproduce the model proposed in the paper of Nestler, Mas, and collaborators called Bayesian computation emerges in generic cortical microcircuits through spike time independent plasticity. And basically in this work, they propose a mechanism in which a simple feed-forward neural network can implement Bayesian inference. And Bayesian inference is something that might be important when you think about processing information, because the brain is constantly measuring noisy data through sensory inputs, and it has to decide which is the actual state of the world given this noisy data. And it has been seen in sensory motor tasks and visual stimuli processing, and even in everyday cognition, that the brain kind of implements Bayesian inference computations, like sometimes optimally, sometimes suboptimally, but it resembles Bayesian inference computation. So now I will explain a bit what is this model about, and then tell you how we implement it. So the idea is that we have a hidden cause, that generates some multimodal distribution in some observable variables. And these observable variables are encoded in a population of neurons. How the brain encodes this information, it's out of the scope of this work. So we assume the population coding of this stimuli. And this population of input neurons project to a second layer, which we will call output neurons, that through a STDP plasticity rule and winner-take-all dynamics, one neuron of this layer learns to fire or to infer which is the hidden cause, which is generating this multimodal distribution that generates this population encoding. So this is the model. And an example of this would be, for example, we have neurons that encode orientation. So for a given angle, the neurons generate some population activity. And through the STDP and the winner-takes-all dynamics, the neuron seed fires every time this orientation is given as an input stimuli. And why am I talking about Bayesian inference? Well, because we would expect that the brain would like to choose this hidden cause that generates this multimodal distribution that maximizes the probability that we are having this hidden cause given this population activity. And because of Bayes' theorem, this probability can be expressed as the prior knowledge of the hidden causes multiplied by the likelihood, which is the probability that we have a certain population activity given the hidden cause. And what Nestler and collaborators show in this work is that through the synaptic time-dependent plasticity rule they proposed and the winner-take-all dynamics, that weights convert to the logarithm of these probabilities, and then they can express this conditional probability of the hidden cause as the probability that only one spike comes from the population output neurons. So this is the main idea. It doesn't matter to go into details of the mathematical formulation of this. So now I will show you which are the main ingredients of this network. Basically, the output neurons receive at each time an input, which is this bias, and the weighted sum of the input population, which encodes the stimuli which is receiving the network. And these population of neurons are modeled as Poissonian neurons. So at each time, each output neuron has the probability to do a spike proportional to the exponential of this input minus an inhibitory current, which enables the network to have winner-take-all dynamics. And the weights are updated following this simple STDP rule of potentiation and depression, which resembles more complex STDP rules we may observe in a real brain. And also the biases are modified at each time following that rule. So well, now I'll talk about how we implemented this network, which was actually really tricky because we didn't have the code available because this paper is from 2013 and the authors didn't upload it because they weren't forced to do it. And some of the implementations, most of it regarding how to implement the inhibitory current that sustains winner-take-all dynamics, were written in prose, like not in equations. So it was, I mean, we had to do some kind of interpretation on how to implement this current. And in fact, they claim in the paper that although this is the optimal inhibitor current you want to apply to a network to sustain the winner-take-all dynamics, that you can model it. I mean, this is not that biologically possible, and they claim that you can model it as a population of inhibiting neurons that interact with output layer, but we couldn't implement that. So we just modeled the inhibitor current as the logarithm of the mean firing rate of the output layer. We wrote the code from scratch and tested the network by encoding 4 different orientations in the population activity, input population activity, which again, we modeled them as Poissonian neurons, and their firing rate, it's modulated by a Gaussian function that depends on the preferred angle, which was selected randomly in the population. We use 1,000 population input neurons. And so what we observe is that by sending for a given time and in a random sequence each of these angles to the network and letting it evolve and learn through the STDP rule, that the weights tended to convert to a stable value. This is the plot of the mean input weight each output neuron receives. And the biases, although they fluctuated quite a lot, they always remain towards a mean stable value. And just by watching the evolution of the input connectivity matrix, one can see that from a random initial connectivity feed-forward matrix, the neurons learn to fire according to each one according to a different angle. Here in the y-axis, we would have the output neurons, which are 4, and in the x-axis, we would have the input neurons, which are 900. And here, these blobs means that this neuron is responding only to the angle that is represented by these neurons. And by observing the raster plots, we can see that before the training, the output neurons don't have any preferred firing according to the angle the network is receiving, which is encoded by the population. But after learning, we see that neurons learn to fire according to a preferred angle, as we see here. So once we had the network working, we wanted to test a hypothesis that was, it just consisted on give a more complex input to the network. So our idea is that if the inputs now come from the activity of grid cells from different modules at a certain place, then the output neurons should learn to fire different place fields. So for those who don't remember, grid cells are these neurons in the internal cortex that fire according to the position of an animal explained in an open field in a very particular way because they fire on the nodes of an hexagonal lattice, which is located along all the open field they are exploring. And they are topographically organized in modules. And within each module, we have grid cells that share orientation and spacing, but differ in their space phases. So the idea was, I mean, these would be the tuning curves of different grid cells with different spacing and different orientations. And the idea was to generate different grid cells with different tuning curves and to feed this to the network. So given a hidden clause that would be the position of the animal in some place of the open field, we would have some distribution of inputs coming from the grid cells. And we would expect that after learning, each output neuron would learn a particular place to fire with. And that's actually what we observe. Like before, output neurons don't fire according to the input position. They just fire almost randomly according to the different codes they receive from the input layer. And after learning, we see that each neuron turns to fire for a specific position. And well, this is all we could do. We could successfully implement the Nestler network, and we were able to infer hidden states of different type of complexities. But we had another objective in our work, which was to implement this network in a more biologically plausible manner. And we plan to do this by implementing it in Nest with leaf neurons. But I think we underestimate how difficult that was, and how difficult it is to generate winner-take-all dynamics with leaky integrator and fire neurons. And I mean, actually, most of the work we've been doing like for the past 2 weeks was to implement this in Nest, and we can't show any results because nothing would turn out from that process. And for example, this is a paper I found 2 days ago, actually, that discusses how, like, it claims that it's not really well understand how to generate these dynamics in leaky integrated and fine neurons. And they proposed that instead of having a winner-take-all dynamics in single cell, to have winner-take-all dynamics in population coding by introducing a structure inhibitory connectivity to another layer of inhibitory neurons, which are centers around inhibition, and that increases with distance. So, I mean, this was the nearest we could get to generating these attractor stages and try to make it learn to the different inputs, but we couldn't manage to make the network actually learn different hidden states. So that's all we've done with the work. Thank you.
When you're modeling the grid cells to place cells, so how are you choosing the position of the animal? So I think I understand that the position of the animal is here at one spot in the environment, and then it's maybe at another spot.
Yeah, so we choose like, in fact, we chose like 1,000 different positions in space. And so for each position, we have a different population code coming from different grid cells which fire differently at each position. Here? Yeah, I mean, it's not the best plot to show this, actually, sorry. But here, you say? Yeah, so the idea is that, I mean, yeah, it's not quite well shown here, but this coding is the same as this, and this is and the same as this. So it represents only one position. And this position is then represented only by this neuron and not the other open neurons. Yeah. I mean, for the 1,000 position, we have 1,000 output neurons. Yeah.
So I think there's so this is, I mean, this is really interesting. There's one tweak maybe that, so the place cells and the grid cells, they really are a description when the animal is moving. And when the animal stops, it actually goes into a different mode where you have the sharp wave ripples and all of this. And so when it's moving, it's actually moving through the whole pattern of grid cells and place cells. So there may be a little bit more complexity in that the spiking pattern may change.
Yeah, I mean, yes, I mean, we didn't intend to explain how places generate, because actually it is not known, up to what I know, that place is generated from grid cells or if it's the other way around. But I mean, if median entorhinal cortex is doing path integration, then you would expect that from the activity of many different grid cells, you could predict in which position you are in the absence of external stimuli, for example. So this might be a mechanism for doing that. But yeah, sure. Also, another thing we thought about, but we didn't have time to do, was to introduce a recurrent excitatory connectivity between the output layer to introduce a time variant input and see what happened there. But it's more complex. Yeah. Yes. Yes. Sure. Yeah. Well, a lot of things. Actually, I learned about a lot about Nest. I find it incredible how with this program you can manage all the complexity you can handle in biophysical models. In fact, I've never worked with like realistic models, like most of them rate models and more like toy models. So yeah, I think like programming things in S, although I didn't show any result because we didn't have anything to show like as a result, gave me a really good insight on how to model more biophysical systems. I of course a lot of biology too because I didn't have background in biology.
Good afternoon, everybody. Okay, so my name is Martina Acevedo, and he is Valentino Ushó, and we are going to present our work that is called Dynamics of Neural Networks of Excitatory and Inhibitory Neurons. So we study neural networks in which the individual neurons were either excitatory or inhibitory. And in particular, we were interested in two possible states of the network that could be a synchronous state in which the activity of the neurons is highly synchronized, and we can see that is highly correlated, and then another state that we call asynchronous state in which the activity of the neurons is not correlated at all, and the events seems to be independent one of each other. So we were interested in understanding what conditions could a neural network exhibit, under what conditions a neural network can exhibit these both states. So we based our study in this paper that both Hansel and Mato in 2003, and in particular they examine how the emergence of these both states when they changing the coupling strengths between the neurons in these networks. So, for instance, suppose that we have two neurons, one excitatory, another inhibitory. As we are interested in the coupling strengths, then if we consider that the postsynaptic neuron is the inhibitory, then we have this coupling strength related, and then the other way around is the postsynaptic neuron is the excitatory one. We have this other weight, and we also could have connections between two excitatory neurons or two inhibitory neurons. And in this model, we fixed these parameters, these weights, to this value, where this little g is a constant, and beta is the size of the population of where the presynaptic neuron comes from. So, with all these connections, we can construct a neural network, and in particular, we are going to use a fully connected neural network. In this project, we use half of our populations to be inhibitory and the other half is excitatory. So we now have the topology of our network, and we have to specify what is the intrinsic dynamic of each neuron. So we use a quadratic integrated amplifier model that is given by this formula. And we say that our neural spikes with the member potential crosses this threshold value, and then at the next time step, we reset the potential to this reset value. So this model was implemented on the paper that I mentioned before, and is particularly interesting because here we have the normal form of a saddle nerve bifurcation. So this model could be a reducing model for any type of one-conducted spacing model, as they have this saddle nerve bifurcation. So, an example that we developed about this conducted spacing model is the One-Bosaki model, in which is a simplification of the Hatching-Huxley model. And we tried to mimic this more realistic model to the quadratic integrated amplifier one. And for that, we changed the parameters involved in the quadratic integrated amplifier model, and we could mimic the same behavior for both models.
So I will talk about a bit from the current that receives our neurons. So, for example, we have a generic neuron that could be of the inhibitory population or the excitatory population. And to that neuron arrive a total current that have two contributions, the synaptic current and the background current. So the synaptic current, we use that model. So for the alpha population, we have the contribution of all the beta populations, spikings, yes, that have these strings, and have the form of the postsynaptic current that is S beta. And this is S beta is the sum from all the spikes of an exponential kernel. That seems in general like something like that. We have the T beta spike, that is the time of the spike, and the tau that it's measure the constant of time of the synapses. So the other contribution is the background current that we will use a Gaussian distribution. So in each time, for each population, we will take one number of this distribution and put that to in the background current. So with this type of currents, we can analyze what is the response of the population, yes? And we can calculate the firing rate of each population. So when we have the last interview with the tutors, they will recommend us to calculate what is the the narrow of that firing rate distribution evolve with when we change the number of neurons from the network. So in this case, we have 2,600 neurons, and we have this firing rate distribution. And if we increase the number of neurons, we have a more stretched distributions. So that's mean that our model is how to say is going well, because that is we expect when we increase the number of neurons. So if we take the limit that the number of neurons is infinite, we will have the delta in the firing rate. So with all of that we say, we can now define how we go to evaluate the synchrony of one state. For that, we use that order parameter that is chi, that have two parts. First, we have the standard deviation of the membrane potential of the whole population, and this is over the mean, the sum of the standard deviation of each neurons. So when this value goes to 0, we have an asynchronic state, and when this value goes to 1, we have asynchronic state. So with that, we can evaluate the behavior of the network. So now we go to the main part of the work, that is try to see the change with one asynchronic state to asynchronic to a synchronic state. So we have that phase diagram that was in the Hansel-Mato paper, and that we will do is we fix the frequency, the mean of the frequency of the population. That we can do that tuning the background current. We put that the standard deviation of that background current is the same for the both populations, and we will work with that number of neurons. So this phase diagram is have only two parameters that they show analytically that the state of the neuron only depends of the determinant of the coupling matrix and the trace of this matrix. So we start in the middle of the phase diagram with a trace equal to 0 and determinant equal to 0, and we then go down with the trace, only changing one parameter, and we want to see what happened when we cross that nucleus. That this one of the old bifurcations that we can see.
Okay, so this is the result that we obtained. So as Valentin said, we started in that point. So that it means that we have the trace and the determinant of the connectivity matrix in 0. And then we started by decreasing the trace of the matrix and fixing the determinant, going downstairs in this graph. So the situation in which we have this determinant and trace 0 is corresponded to this state, and we can see that when we are like incrementing, decreasing the trace of the matrix, we are going down in this graph. We are still on the asynchronic state, but there is a point in which this measure, that is the order parameter that we said that is 1 when we have this synchronous state, and 0, we have this asynchronous state, is increasing. So this means that our population is getting more synchronized. So, for instance, we only studied this this transition, but we wanted to also move in other directions of the phase diagram, but we couldn't obtain the expected results because we didn't use exactly the same parameters that were shown in the paper. We use a more simplified model. So we think that the shape of this asynchronous state is maybe a little different, so we couldn't reproduce the other phase transitions that we tried. So, in this work, we utilized a quadratic integrated amplifier model with the parameters derived with this one Buzzacchi conductor spacing model. And we verified the dependency of the firing rate distribution with the distribution with the size of the network. That was what we expected.
And at all, we explore synchronous and asynchronous state across different coupling strings with the network, and we can identify the emergence of a synchronous state in the specific direction of the phase diagram. So, future directions, if we continue in this with this work, we want to try to analyze the other transitions, because sometimes, for example, if you try to see this transition, the problem is that is in our case is so near the start point that we take. So it's difficult to see the transition. So if we, with more time, we can try to describe that type of things. And we talked with Tilo the last week, and he said that that type of networks with quadratic integrated amplifier neurons, if we put a specific background current, we can solve analytically this type of problems and have a type of a macroscopic model. So the idea is if we could try to change the noise, the background current, and compare with that analytical result. And the other thing that we, at the start of the project, we want to do is change the type of the neuron to see the difference change in the phase diagram and the transitions. So, thanks.
Yes. The frame rates are a bit large.
The frame rates?
I don't know if the axes were correct or not, but you had something like 50 Hz.
Yes. Yes. But
a bit unrealistic.
Yes, in the paper they use 50 Hz too. Yeah, between 30 and 50. And they use this current that maybe the axis is too low, but is around 0.5 microamperes.
Changing with time constant, it would be more realistic.
Okay.
Is the one that the person correctly matches.
Yes, that's all. If you can tune that, we can tune that, changing the background noise, the background current. So if you want, you can put in 30 if you want.
Also, I think you mentioned that you guys considered half of the network being excitatory and inhibitory. Can you comment a little bit more and explain why you choose that?
See, I think that they want a more symmetric situation. For example, that phase diagram that we put in the presentation is the most symmetric case. On all the things are symmetric. So in that way, I think that they take that for simplicity. Yes. Yes. Then I don't see they in some part they changed that relation, but they, for example, changed the sigma of the populations, so they start to appear more strange things that dot point line start to be another bifurcation and things happens.
It's very, very elegant. Step by step.
Okay. Well, thank you.
Okay, so when Horacio gave his lecture, he used this dynamical system theory to understand a two neurons problem, and I get interested in applying these dynamical system tools to neuroscience because I see them only in physics course. So I find it really interesting, and I think that it's very useful, and I really enjoy it.
I'm agree with that, and I think that the work to try to reproduce the paper is really nice too. But because we when we start, we say, okay, it's probably will be easy, and we do that in 1 week, and then we can do other things, and that's not the situation. So it's a good experience to try to reproduce something, and I think that is when we will publish something, I think that we have a better view of the thing that we need to put in the paper. Thank you.
Good afternoon, everybody. My name is Hernan Villota, and she is Carla, and we're going to talk about our project. In general, our idea is related with try to evaluate the effect of modulate a specific molecular pathway. This in a context of synaptic plasticity computational model. So for this, first of all, we need to understand why we have interest in this specific molecular pathway. The WNT pathway have a very critical or key role in embryogenic development because his function is related with cell division and actin cell proliferation, right? And it's for this reason also that in cancer research, they have a special interest, for example, find a specific molecular targets for a therapeutics process, right? But here in the context of the brain, we have some studies that indicate the relation between the WNT pathway with Alzheimer's disease. So some studies describe different mechanisms, but we only focus in these mechanisms where the WNT pathway active or function as an inhibitor of an precursor of alpha amyloid beta. That is a very studied molecule in the context of Alzheimer's. So for understand the model, we need to understand in general how this molecular pathway works. So first of all, we have in left the in-off state of the pathway, and in the right, the in-off state, right? And we have three key elements in the pathway. First of all, in orange, the WNT protein that is the inductor of the pathway. The second one, in green, beta-catenin, that is the precursor of the pathway. And finally, this complex of different proteins that have a function of regulate the pathway context. So in the in-off state, we don't have inductor, we don't have WNT. So this complex of proteins is named beta-catenin degradation complex. So its function is degrade the beta-catenin levels in cytoplasm, right? And in the on state, we can see how we have WNT. When WNT bind with this specific receptor in cell membrane, the degradation complex of beta-catenin is inactivated. So beta-catenin increase the levels, start locate to the nucleus, and the nucleus, they acting as a transcription factor. When it start to increase the levels of proteins to stimulate the cell division and cell proliferation. But in Alzheimer's disease, the case is that, next, please, yes. When we have an activation of the pathway, we have this inhibition of these genes named BACE1. So this gene is a precursor of alpha amyloid beta oligomers. So in Alzheimer's, this alpha amyloid beta is related with decrease the gene expression of the receptors, NMDA receptors, and decrease the conductance of these receptors too. So the idea is try to modulate the behavior of this molecular pathway and in the in-off and the in-on state. So this is our objectives. First of all, develop a computational model based on differential equation for try to represent the dynamics of the pathway. After that, verify and validate the model if have concordancia with in vivo or in experimental evidences. Finally, implement this pathway model with the synaptic plasticity model and try to evaluate what happened with the LTP levels when we try to modulate this pathway. So for the methods, first of all, we only focus in four elements, five, the WNT protein, HCK3 beta, beta-catenin, el gene BC1, y alpha amyloid beta. We implement with differential equations. So for example, in this, you can see that the levels of WNT is dependent of the synaptic synthesis and synaptic degradation of the WNT. For example, in the case of BACE1 gene, this is related with this equation of Hills that try to modulate the cooperativity process between two molecules in this case, and also the same with the outer elements of the pathway. And after that, we implement the model.
So basically, our code and all project was based on a paper on 2023 of Dainauskas et al. And basically, they use a two-compartment Pinsky-Rinzel model of CA1 neurons, which has a somatic compartment coupled MDA receptors containing subunits on LTD and LTP influence, respectively. So basically, how we calculate the net weight change of the synaptic net weight change. We sum the MDA R-dependent LTP and LTD weight contributions, which are proportional to the phi MDA either LTP or LTD functions multiplied by a low pass filter postsynaptic membrane potential. And also, like, it is important to know that this phi NMDA function, either the LTP one and the LTD, is described as a Hill equation, and also thresholded by this moving threshold function theta, that this will allow us to have the competition between LTP or LTD. And well, basically, with the past literature we research, what we've found is that the WNT amyloid beta pathway, alterations in this pathway leads to alterations specifically in the GLU N2V contain subunit, receptors containing this type of subunits. Yeah. For the results in general, from the pathway that my partner explained, we have this dynamic of the WNT amyloid beta pathway and its impact on the MDA receptor conductance, which is here, at varying concentrations of WNT. So we can see that when the synthesis of WNT is low, we have a minimal conductance of GLU N2B, because amyloid beta is present, so it's like altering the conductance of these type of subunit receptors. And when we have big concentrations of WNT, we can see that the conductance it's bigger than with lower concentrations, high concentrations.
Yeah, thank you. So now I try to explain the previous data with graphics. So first of all, we have here the behavior of the beta-catenin protein. So this is the behavior of our model that, if you compare with experimental evidence, we show how, for example, in this case, the black region is the quantity of the proteins. So here we have how, if you increase the levels of WNT, you increase the levels of beta-catenin in a linear way. So our model describes this behavior. After that, what happened with the GSK3 beta, we have the skull here is very short, and the behavior is like a very stable. So in the experimental data is the same behavior. You have a lot of differences in the concentration of WNT, and the effect of the GSK3 beta is less, right? After, in the context of the gene expression, in this case, the purple line is the gene, BAC1, and the red line is the protein. So here we compare with in vitro experiments, where if you put WNT, decrease the levels of expression of BAC1 gene, and in presence of WNT, decrease the levels of alpha amyloid beta 2. And finally, the effect of the conductances, when you increase the levels of WNT, the conductance increase. So some reports respond the behavior of our model. When we have alpha amyloid, for example, the LTP, for example, in these graphs is modulated, right? So after confirm that our model of the pathway works, we try to evaluate the effect of on the synaptic model.
So basically, we analyze the synaptic weight change on different WNT levels, from low to high, and we had this LTP protocol to confirm that we have that we are able to increase synaptic weight and see the changes relating to different concentrations of WNT. So basically, we have here 4 plots: the membrane potential, the MDA conductance of either glue N2A subunits or glue N2B subunits. I'm sorry, al revés. Yeah. And also the MDA are dependent functions, which are the phi functions that are thresholded with this moving threshold, and the synaptic weight. So basically, what we found is that when there are low concentrations of WNT and also like these high concentrations of beta amyloid beta, we found that with this LTP protocol, like the somatic potential and also the dendritic potentials don't fire so often. So they are not like, they are not responding to this high frequency stimulation. And also we can see that their conductances are low, and the phi function for the LTD component is low and like wins the competition between the LTP function. And also we can see that the synaptic weight like really doesn't change or is low, the change in synaptic weight with low WNT concentrations. So, yeah. So and also when you increase the WNT concentrations, we see that the neurons start to spike and start to respond to the LTP protocol, and we see this increase in the conductances, and also we can see here that the phi function for LTD first like wins the competition, but when we apply the LTP protocol, the phi function for the LTP contributions starts to win it. So we have this increase in the synaptic weight change.
Yeah. Okay, thank you. And finally, when we try to develop the project, we find another report when we evidence how the WNT pathway also can influence the AMPA receptors. So we try to, again, modulate the pathway, but now we compare this condition in 30 nanomolar and 50 nanomolar, and plus the effect of WNT in the context of AMPA contribution. So we can see that the effect of the WNT over the context of AMPA is the decrease the synaptic wave, for example.
And also, like what we found is it's interesting because it's delaying the response of the neuron. Like you can see here that in 30 nanomolar, there are it's responding to two spikes, but with this AMPA modulation, that it is delayed. And also here it's more like visible because the net weight change also is delayed, and also the spikes generation is delayed in general. So like it doesn't like contribute that much to how much synaptic weight is changing, but it's like a delay in getting to what we expected to.
So the conclusions of our project, first of all, the impact of the WNT modulation on synaptic plasticity. We can prove that this implement of this first molecular pathway can describe like an in silico validation of the in vivo reports with these specific parameters like the conductance of the receptors for the NMDA and AMPA. And finally, the objectives of the future perspective is try to introduce more data or complex of this pathway for try to understand in a better way what happened with this regulation process. This is all. Thank you so much. No. The differential equation we try to modulate using the kinetics of the rates of synthesis and degradation of each element of the pathway. Yeah, yeah, yeah. No, they don't use the pathway. They only use the model specifically. But the same authors have another report more recently that they use only the specific modulation of alpha amyloid beta. Apply the modulation in the conductance of the same receptor. Yeah. The tools? Yeah. We implement, the model was implemented in Python and buys it in Neuron.
The two-compartment model was in Neuron and did the rest in Python. Okay, thank you.
I major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there were some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s, Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
I'm doing my PhD in physics, specializing in computational neuroscience.
I did my major in biology, and I'm really entering into a more quantitative field, so I think LASCON is a good experience for me.
So we'd like more people to be involved working in computational neuroscience. We think it's very exciting. It is very exciting. Nobody understands really very much at all about the brain. The brain is important. We can all agree on that. And so if we could understand stuff, we'd be happy. So I encourage the students to think very liberally, think new thoughts, try to do something very different than what I've done, what the other teachers have done.
I had this dream of having a school here in Latin America because there are some Latin American students attending those schools in Europe or US, but there are few. I know there are more people here. There's a kind of repressed demand for that in our continent. So actually in 2005, I believe, I was in a conference like that with a guy who was the organizer of the first American school like this in the 80s. Jim Bauer. So he put me in contact with people who work on programs, neural simulators, and then I invited them. They came to Ribeirão Preto, to Brazil in 2006, and then we had the first one there. And then I noticed that it's possible. Actually, it's possible to do something in Brazil which works.
Here they're working in teams, and so they may be able to combine their expertise, one from biology, one from physics or math, and try to build something together. And that's, really the best way to learn is through hands-on experiences.
Hello, hello, hello. Hello, hello. Is it working? Yes. Yeah. Okay. Good night. Good night, everyone. Thanks for staying until this hour of the day. Must be very tiring. It's almost over. I'm happy to present here today our progress in our project, which is related to the dendritic formation, the dendritic growth in these particular and special cells located in the hippocampus. Let's see. First of all, let me talk a couple of minutes about the context of this project. We are working mainly on the temporal lobe epilepsy model, which is a very well-known model for epilepsy, in this case, recurrent focal epilepsy. And there is a lot of information, both from humans, from rodents. One particular feature of this model is the following. We have here in the dentate gyrus of the hippocampus these tiny cells, these excitatory cells, which are the granule cells, that goes through, neurogenesis. That is, newborn cells comes from this, what is it? Okay. From this layer, which is the granule layer, and develops, mature, and casts their dendrites to this molecular layer, reaching the perforant pathway coming from the entorhinal cortex. This is in a normal situation. Now, in an epileptic hippocampus, we observe that these new born granule cells tend to cast their dendrites to, or try to reach this peripheral pathway, but they are immersed in this situation in which there is a lot of changes, both from the tissue and in the electrophysiological properties of this layer. So when they mature in this time, they also tend to cast aberrant dendrite to this same layer, producing connections between other excitatory neurons present in this layer and making a recurrent circuit, excitatory circuit, as it can be seen also in this kind of images. So this is a problem. Now, to study this, there has been a lot of information studies related to this process. Now we are focusing in the computational studies in these kind of situations. One of them tries to relate the morphological properties of these newborn neurons in this context of epilepsy, and try to, of course, test some hypotheses in this model, this dendritic model, this morphological model, which is trying to do some research to study some questions about the electrophysiological properties that are related to its morphology. This article was published in 2012, but very interesting because it gets about a bunch of data from morphological granule cells, and they reconstructed it, and then they tested some hypotheses here. 2 years later, this article was very interesting because they put this model, this morphological model in a circuit, properly speaking, in the dentate gyrus. Basically, what the authors did was to emulate how these neurons produced these connections between granule cells and what is known as mossy fiber sprouting, and they evaluated the main electrophysiological properties of the whole circuit. So in this case, I wanted to point out this because it is very interesting to know that the morphology of these cells can explain, in some sense, the later electrophysiological properties of the circuit. So we have these images, these simulations, but then we came into a question, a really interesting question for us, that was, yes, we have this information from experimental data, we have these models, but when we search information about these models, even in the classes, we saw that this morphology was already fixed. That is, when you search in the literature, we see a fixed point in which the neuron was already developed, both in a normal situation and an aberrant situation. And we wanted to know how this happened, how from an immature granule cell come to this place or to this place depending on the surroundings. Okay, so we searched for a lot of models, computational models, and to be honest, the majority of the articles trying to explain the morphological formation try to explain it through a probabilistic model in which, based on a probability, it can branch or it can elongate. That's an interesting point of view. Professor Arn suggested us 2 articles that was published just the last year by the Professor De Schroeter, and it was very interesting to read. However, we know that from a biological point of view, this situation can be due to different factors, such as, for example, the early depolarization of the interneurons produced by the interneuron, the GABAergic neurons. You know, the tubulin polymerization that is related to calcium signaling, and also the excitatory synapses in a process of maturation when these neurons reaches this area. So there are a lot of biological causes that can be producing these two scenarios, no? Isn't it? So the main question was this: what factor determines the dendritic growth of a newborn and granular cells in the hippocampus? For this situation, our first objective, that was the objective in which we focused on this month in the LASCON, was to try to develop, try to establish, to try to run a probabilistic model that was firstly published in the literature, and then for future studies, we would like to try to implement those other factors that I have mentioned it before, to do this probabilistic, to do these factors, to make these factors affect this probabilistic model. So we reach one of these conditions depending on the presence or absence of these factors. So now Adriana is going to tell the second part of the story.
Okay, thank you. I'm gonna be a lot less biological, but I hope that you like some nice pictures. Okay, so to start studying the morphology and the evolution of the morphology, we try to implement ABM's models, agent-based models. And our first and basic structure will be like this soma, an agent soma, principal compartment, an intermediate compartment, and a terminal compartment. So as my partner was talking, we will focus just on the branching probabilistic model of to see the evolution of the morphology. So we are just gonna focus on the terminal compartment and evaluate on that last agent, temporally terminal, the probability of branching and the probability of elongation. So our first thought and our first plan to do all of this is first simulate a control neuron that will have NI inputs and a certain depth of glandular cell versus a neuron on a pilocarpine model that will have an NJ inputs, and it will have obviously less inputs than the control neuron. So our expected results will have implied having the comparison of the complexity, the dendritic complexity versus the number of inputs. So we'll just expect to have NK inputs versus the dendritic length, and we'll expect to have some certain correlations. And then also we will try to compare the number of branches that appear in the morphology versus the number of inputs that our neuron will have, just being the control or the pilocarpine. When we proposed this project, we were advised to use the neurodesign that was published just on this paper of the Schroeder in 2022. But after fighting with the installation and all that, I just desisted because it only runs on Mac, so I was really fighting with the virtual machine. But what we had, the other option that we had, was using Repos4Pi that also allows for ABM models with Python. And another thing that, something good that happened with neurodeep science is that the documentation is free, so we could base some of our basic algorithm on the documentation. So this is something a lot similar from what I call the basic somatosensory compartment structure. So in neurodeep science, they also start with this kind of model. So they start with 4 cycles. That is first the soma extension event, the extension event that extends the soma, And then two events, that is what's important for us. That is the branching of a compartment and also the elongation of the compartments. So tests or algorithm, we first put a naive probability, a constant probability of 1/2, so we can check that our algorithm worked. And this is what we got. This is the branching evolution of our basic algorithm. This is after 500 iterations, and this is just after 10 iterations. So we got really nice things, but this is nothing, I think, biologically real. So yeah, but this is just to test that our algorithm worked. And then, obviously, coming back to Earth and thinking about how are we going to implement and do the control neuron and the pilocarpine neuron, we have to analyze more about morphological analysis and modeling of neural dendritics. So we came across this paper where they present three models. The first model, that is the QS model, B model, BS model. And basically what they do is just classify the evolution of the dendritic morphology from topological and degree. So they start presenting these parameters of their models, that is order, that is like the layer of the branching, and then the degree, that will be like the number of branches after a branching process. So the first is the QS model that has the probability of the terminal segment to branch, then the probability of the intermediate segment to branch, and this is just the normalization constant that they use. But we don't really care about the branching of the intermediate compartment, so we just ignore it. And it is also advised by the authors that we can just put Q equals 0 and go on with your life, because it doesn't really matter. We're just gonna focus on the terminal compartment branching. So yeah, something that happened here is that we got, after only 10 iterations, we saw branching evolution really critical. It's really disordered. So we thought, why is this happening? So we analyzed the probability function of the PV-PTS, and we saw that it was always 1. Okay, so yeah, this doesn't really matter to us. So we went to the other model, but we didn't also care about it, because what was said in the paper is that they gave the same probability to every branch, to every last point. So every last point had the same probability of branching, and that was basically what we did first. So we didn't care about that. And we passed to the BES model, where they just focus on the branching of the terminal segment, and they also add 2 parameters more. That is the expected number of branches and the strength of branching probability of a terminal that depends on the actual number of segments. So this will help us to manage and control more the evolution of the dendritic morphology. And we got some, this is the nicest pictures that we could get. And also, we run this 100 times, and this is all we got, because the probability function decays rather quickly. So for our next steps would be implementing the number of inputs or the granular length to make this equation, this probability decay rather slowly, so it functions as we all want it. And yeah, for some future work, as you can see, we didn't got the full expectation of what we wanted to compare the control and the pilocarpine neuron. But we would like to look forward for the implementation of this model that can generate a proper morphology and study the pilocarpine regime, and also make a statistical analysis of the generated morphologies with our model and compare it to real data. So yeah, that would be it. Thank you.
Branching model, it's something that you learned here in LASCON?
We talked with Mauricio, and he told us that he worked with some branching models, and we talked a little bit about it. So I just started looking for it. Yes. Yeah, we first started simulating the voltage transmission towards the dendritic compartments using Cabel equation. That was like the first part of the project, but then we turned into this. But what I liked most and what I learned most about computational neuroscience here, it's the amount of things that you can do, and also I really like that there are a lot of physicists here. So coming from a physics background that doesn't have this, that doesn't feel the biology theory thing, I really like it a lot, and I learned a lot of biology here. Yes.
Yeah. Yes. If you see the program, maybe there is not too much classes directed to talk about morphology, and especially in this case of studies, because we are trying to study this phenomena using an agent-based model, which is not too common, at least in LASCON, we haven't seen that. But in fact, we have learned a lot of professors, which also work with morphology. In this case, Professor R, in his works, he showed us a lot of his works in Purkinje cells, which is very interesting because they are combining both morphology and electrophysiology. This is one of our goals for a future work was to implement this analysis of the electrophysiology in this model of morphology, because in this agent-based model, we have the luck that every branch is considered as a compartment. So it's kind of easy to implement this kind of situations here. And also we have the other hypothesis we wanted to test, especially on those probabilistic equations. And we want to see how these probabilities changes and goes more for a ramification branching or a longing, depending on, for example, the presence of factors such as interneurons, giving this depolarization on these young granular cells.
Merge with group 3.
So
you should put them even there, the branching models, and you put the rest.
Yeah, it was a very exciting experience, really humbling, because for me, who comes from a biological background, I realized that there is a lot of things I don't know, and I am really, really eager to learn, especially from the mathematical and computational perspective. I think it is very important for everyone, at least from those who come from the biological background, and I am very happy for that.
There? Okay, so good afternoon, everyone. So I'm Alejandro, he's Andres, and together we'll present you our project titled NetPyNE Exploration of Hippocampus CA1 Microcircuit Towards Epileptic-like LFPs, although maybe it should say activity in a more general way. So first, a little bit of contextualization. So epilepsy is the most common neurological disorder, and is mostly characterized by presenting these spontaneous recurrent seizures, right? So seizures are these symptoms caused by abnormal synchronous neural activity, right? But actually the story begins far more before, with the so-called initial precipitating stage, which might have very different causes. And right before this stage comes the so-called latent phase, which has a high variability of the duration and no typical behavioral output. And after that, you actually develop the chronic phase. So that would be for what is epilepsy. And so we focused on temporal lobe epilepsy because it is the most common type of epilepsy, and the name comes from the part of the brain where the epileptic activity comes from, right? So in the temporal lobe, we have both the amygdala and the hippocampus. Both are structures that inherently are really excitable structures and usually have patterns of activity of synchronous activity. So yeah, we focus here on the CA1 part of the hippocampus. And so the actual paradigm is, this is relating to the hippocampus. So in the hippocampus, we usually have, as I was telling you, high frequency oscillation events, like the sharp wave ripples, which are important for memory consolidation during sleep or quiet wakefulness. And the thing is that after the initial precipitating stage that I told you about, literature says that you can actually see these biomarkers that are, you can see them as a biomarker of epilepsy, right? Like the preparing stage, the latent stage of epilepsy. So you would see high frequency of oscillations, right, as in the physiological state, but these high frequency oscillations will have a higher amplitude and will have a higher probability of occurring. So with all that said, the idea of our project was, first of all, to model a CA1 network, and after that, tweak parameters in order to simulate an epileptic-like CA1 network. And this would be all to identify eventually mechanisms that would explain why this network is showing this epileptic-like activity.
So now let's delve in the methodology. So initially we found this model in ModelDB from by Turi et al. 2019 that features 7 neural populations, 1 excitatory pyramidal cells, and then the other 6 populations, 6 inhibitory populations. We decided to reproduce and mimic, well, replicated this microcircuit in NetPyNE. Initially, we also found the repository, this model by Neymot et al., who modeled these kind of configurations of 3 neural populations: the pyramidal cells, the basket cells, and the OLM cells. We also, we started with this template, and then we expanded to the 7 populations that we saw previously. So we imported the parameters, the morphologies for 5 of the 7 populations. There is a little constraint that 2 of the populations, we created the morphology as a single point neuron of just 1 compartment because the file didn't work well. So that's a limitation of this part of the model. Basically, also we roughly estimated some of the parameters like the convergence, well, the probability that some kind of populations connected to the other populations. So, and we also selected some arbitrarily values to other kind of missing values, parameters, in order to make the network similar to the, or in the more realistic way, even if it is not the best one. Then, well, after performing the simulations of the previous microcircuit with 200 neurons, you can see here a power law graph which has different peaks, mainly these two first ones at low band frequencies. This is the normal state of that we take as the control of this simulation. Also, we perform before that, this PSD is from the local field potential, because we recorded this local field potential in order to facilitate the search of high frequency oscillations biomarkers, because it's more common to find these biomarkers at the LFP level. Also, this is the spectrogram of that local potential. You can see a kind of synchrony at the first 50 milliseconds, but as Professor Battaglia mentioned this morning, well, we think it is like the initial values that just at the beginning are random and then converge. So, well, now we think these are not the desired results, but well, that's the results that we got previously. And this is the raster plot. So maybe we can see like a coupling between the inhibition populations and like the initial coordination of pyramidal cells. But well, we have to improve that. For future work, we will work on that part.
Right, so from there on, we started tweaking, as I told you, some parameters. So the first parameter we focused on was on modify the tau values for the AMPA currents at the network level, at the whole network level, not in a specific cell. And we see that in the power spectral density plot, that actually the high frequency component does increase in comparison to the so-called physiological state. Also, in the spectrogram, we can also see some sparse lines on the high frequency component side, but these ones are very short-lived, right? So the simulation only lasts for 700 milliseconds. And finally, here's the raster plot for the tau values, and we can see that even though the mean firing rate of the neurons is lower in the so-called epileptic-like state, the synchronicity does increase in comparison to the physiological state. So that was one experiment. Then the next experiment we did, which is an independent one, was to reduce the potassium conductance in pyramidal cells, as would happen maybe in some mutation. So in this case, the power spectral density also kinda increases in the high frequency components. Well, and in the spectrogram, it does not look really like something you could easily read maybe. This should not be here, I'm sorry. And finally, the raster plot of the epileptic-like state does show an increase in the mean firing rate, although it lowers the synchronicity. So yeah, that would be our two simulations.
So to finish, we want to conclude that initially we saw like this change of the parameters could derive or could affect the whole network in a particular way. So we have to focus on effects of the lower conductance and affectations in the AMPA channels. But basically, we know that it affects the network, but we need to refine it in order to conclude any relevant thing. And for future work, well, refine the experimental values for parameters through testing and adjustment, enhance the self-morphologies. As I told you, there are some neurons that some population of neurons that are modeled like just a single point neurons. So we have to add some compartments to them and increase the complexity of that dynamics. Also conduct the comparative statistical analysis and perform a comparison with experimental data. And yeah, that's it. We want to thank Valerie for your patience and your guidance on this project. And yeah, of course, Professor Roque for this school, and the team LASCON for every class, you know. So, I mean, and that's it. Thanks.
There's a change in state with, if you look at your raster plots in the normal state.
This one?
Yeah, that's good. And then, so you look at the physiological state, the epileptic-like state, there's a change in state specifically with a couple of the cell types. Could you say a little bit more about that, or is there something interesting, and maybe could you relate that to how you would compare it with experimental data?
Yeah, we think that probably, for example, this lower potassium conductance would lead to a high spiking of the neurons, to a higher frequency. But we don't want to conclude about it because the whole network doesn't work properly. So we know, because we based on some papers, that probably this parameter controls the high frequency in the epileptic-like state. But yeah, it looks like promising work if we can get a functional microcircuit.
There's something extra too. If you look at the basket cells, they go into a counterphase relationship with the rest of the population, while the axo-axonic cells don't. So they are mostly the same, and then the basket cells are counterphase with the rest of the population. So you could actually try to look for that experimental.
Okay, sure.
Effectively on the OLMs.
Poor OLMs, yeah.
OLM cell bursting that inhibits for the rest of the
Yeah.
The yellow.
I know it's BCA.
Okay.
It seems it was in the other raster plots.
Yeah, should there have for some Rosa Cosara or Christophe Bernard on the inhibitory hypersynchrony in epilepsy, which may make the link. Yeah, maybe it's not totally wrong.
Well, we will see. Okay, that's all. Well, before I came here, I was a little bit disappointed of the biophysical single-cell models, but then I came in and I learned, for example, that we can do this kind of multi-scale analysis and we can extrapolate to more network populations and to the higher complex dynamics. So now I want to study from the lower level to the higher level and learn of all of these tools. For example, for NetPyNE, I didn't, well, I think that if this project was give the, the properly results, probably I didn't go to the source code and see the functions and learn from that part. I guess that maybe the mistakes are the way to learn, and it is very important to make some errors in order to correct in the future. So I learned theoretic things and, well, that kind of things that will help in pursuing, I don't know, a PhD or a postdoc or whatever.
No, for me, so, I'm a biologist, so as others have already stated, I don't know programming like at all, and math as well. So in that sense, it was really a challenge, but still it was nice to know that all these tools exist, like NetPyNE, Nest, Neuron, and that the model field is something that is really there, because I didn't even know that it was actually a thing. So for me, I'm happy with that. Yep.
Thank you.
Ask the students to leave the room, please, because we have to discuss. If you need some interval before that, it's okay, but we'll discuss, there's a prize. So we have to discuss about who is the winning and the honorary members. And I'll call you. Yeah, no spying. Yeah. And for the ones who are watching via YouTube, I think that's the end. We'll not, yeah.